Thank you for viewing my talk. My name is Marko Spasojevic, and I'm an assistant professor at the University of California, Riverside. Today, I'm going to talk to you about managed relocation and the importance of considering species interactions and relocation efforts. I'd like to start by acknowledging and thanking my co-authors who helped make this work possible. One key question in conservation science is how and if species will survive climate change. Some species will have the ability to adapt by natural selection and evolve in the face of climate change. Others will be able to disperse and move to more suitable habitats. And some will simply have the stress-tolerant traits that allow them to survive and persist given climate change. For the species that do not have one of those three strategies, one conservation strategy that has been widely discussed but rarely implemented is managed relocation. This is any type of conservation management in which at-risk species are intentionally moved to promote their persistence during rapid climate change. This idea has been around for a while, but relocation frameworks early on really focused on climatic suitability and on single species in isolation. This example here shows modeling the potential habitats for the Asian elephant in North America, right? And so this wasn't really going to happen. A lot of the early work also focused on the effects of relocated species on recipient systems. A lot of criticisms of whether or not managed relocation is possible or not. What I want to focus on today is not a debate into whether or not managed relocation should or should not be undertaken, but when it is, because it is happening, that species interactions need to be explicitly considered, as these will play a large role in dictating the success or failure of any attempt. So today I'm going to focus on managed relocation of plants. So here in my red box is my target species that I'm relocating. And when we move this into a recipient community, we need to consider the roles of multiple species interactions, such as competition with individual plants in that recipient community, or possibly facilitation, that the recipient community may benefit that new species in that recipient site. When you think about consumers that may consume this plant, the need for pollinators in this new site. And what I'm going to focus on today are soil microbes. All right, so moving a plant to a new site may mean a loss of mutualists, and so it may do worse. Or it may also mean escape from pathogens, and so it does better. And so to exemplify the importance of species interactions, I'm going to share a case study today involving the relocation of the serpentine endemic plant, Horkelia sericata. For those of you unfamiliar with serpentine soils, this is oceanic crust that's uplifted through continental crust, giving it unique chemistry. And because of this unique chemistry, it's a biodiversity hotspot. In this picture here, you can see in the foreground a serpentine grassland full of beautiful wildflowers. And in the background, you see a highly invaded grassland on typical soils that are not serpentine. Additionally, serpentine tends to have an island-like habitat distribution. So here's a map of serpentine in the state of California in the United States. And you can see that for a species to survive climate change that's restricted to serpentine soils, it can't simply just move north because there may not be serpentine soils to the north of it. However, a lot of the serpentine is found in topographically complex landscapes. And so in these topographically complex landscapes, short distance movements may buffer species from climate change. So this figure here is from Scherer and Corner in 2010. And it's showing thermal imaging of this alpine landscape. So here you can see huge microclimatic variation across this landscape. And species may be able to move from simply a red spot to a blue spot to be able to survive climate change. So back in 2011, we established a study looking at the potential for topographic managed relocation of our study species Horkelia. The study was conducted in the Klamath Mountains of southwestern Oregon in the United States. This has a Mediterranean climate and has warmed about 2 degrees Celsius over the last 60 years with no significant change in precipitation. And in this landscape, we set up an experiment where we collected seeds at these 3 sites here at the bottom on a flat aspect, so they're gray, and then planted them into paired north-facing aspects on blue and south-facing aspects in red along an elevation gradient. We also had 1 high elevation site that was flat. This will be important later. At each of these sites, we set up 10-meter square plots that we then subdivided into 4 treatments: a control, a neighbor removal, where we removed all the neighboring vegetation, a litter removal, where we removed all the litter, and a treatment where we did both. We continued these for 2 years, but did not continue them after that. And so along our elevation gradient, we found that the microclimate was quite different. And so this graph shows elevation on the x-axis and temperature on the y-axis. And here we see that minimum temperatures, measured over a year, every 2 hours with eye buttons, did not really differ along this elevation gradient. We start to see a bit of a difference in the mean temperatures. And so those red sites, those south-facing aspects, are a little bit higher. But this difference is really quite different when we look at maximum temperatures. So you can see here that the maximum temperature of those north-facing sites is much, much lower than those south-facing sites. And at this highest elevation location, that difference was about 7 degrees Celsius, which is the equivalent to an elevation gain of about 1,000 meters. Right, so that's a big elevation gain, and those 2 sites were only 450 meters apart. Right, so there's great potential for these north-facing slopes to serve as refugia. So what did we find back in our study? After 2 years, we found just that. Here again, we have elevation on our x-axis and population success on the y-axis, which is measured as the summed biomass of all individuals averaged across plots for a site. The dashed line that's vertical there represents the current elevation range of Horkelia. It grows below that line. And we found that Horkelia did best on north-facing slopes and that flat site above its current elevation range. Also, we found that neighboring plants facilitated those relocated individuals in cooler sites. So again, here we have elevation on our x-axis, and on our y-axis, we have a relative interaction intensity. And a positive value here indicates facilitation, that these relocated individuals grew better with a neighbor than alone, where a negative value indicates competition, where individuals grew better alone than with a neighbor. And we can see a shift from a little bit of competition at low elevation to a fair amount of facilitation at the high elevations. So we did this back in 2011. And after 2 years of my postdoc, we were out of funding, and I moved on. And then once I got a faculty position, one of the lingering questions I had was, well, did this work? So 6 years later, I went back once I got a little money and resurveyed these populations. And for most sites, it didn't work. So you can see here we have a lot of zeros along our elevation gradient, right? A couple sites where there's a 1 or 2 individuals that persisted. But that one high elevation flat site, we actually had 60 individuals that survived from 2011. 4 of those were in flower, and 33 of those produced an additional 83 clonal offshoots. And you can see here in this photo, our string marking our plots was still there after 6 years, and so were some of our toothpicks that we used to tag individuals. And here's a slightly blurry photo of one of our individuals and the 2 clonal offshoots growing off of it. And so one of the questions we had back in our original study was, what was the role of microbes in this relocation? Do they help? Do they hinder? Does the recipient community have an impact? So that was the question we wanted to ask here. What role do soil microbes play in the success of this relocated population? Do they differ between the home population where the seeds were collected and the relocated population? And does the rhizosphere soil at the relocated site differ from bare ground at that site? So does it differ from the home, and is it having an impact on that site where we collected it? So a couple of my colleagues came with me when we did this resurvey, and they collected soils from the rhizosphere and nearby bare ground, sequenced it for ITS and 16S to get fungi and bacteria, and then did all the bioinformatics magic that they do. I'm a plant community ecologist, so I don't really do that stuff. And so here we found that the bacterial communities of the relocated population differ from home, but don't differ from bare soil of the recipient site. So this is a principal coordinates analysis of bacterial composition. Here are the black squares, we have Horkelia at home. The black circles, we have the bare ground at home. And you can see they're significantly different here based on a permanova. Our Horkelia at the recipient site is also different from home, but does not differ from the bare ground at the same site. We found a few differences in bacterial groups between the home and recipient sites, and these groups here highlighted in green. And we found a very similar patterns with the fungi as well, where the Horkelia at home significantly differed from the bare ground at home. Horkelia at the recipient site differed from home, but did not differ from the bare grounds at those home sites. And here we only found minor differences in the fungal groups. There were small, marginally significant trends that overall led to that overall shift in composition. So did the microbes play a role in the success of this relocated population? So the microbial communities do differ between home and the relocated population, but the rhizosphere microbial community of the relocated population does not differ from the bare ground. And those differences we saw to us suggest no evidence of loss of mutualisms or escape from pathogens, and that these interactions might be relatively neutral. We also saw no impact of the soil microbes of relocating Horkelia to these new sites after 6 years. So it seems like there is not an impact on the microbial community of moving this plant. And so what this suggests to us is that rugged topography can provide a really good opportunity for short distance relocations, right? Species interactions are going to be really important in our managed relocation efforts, and this may minimize those novel interactions. In our case, we found positive horizontal interactions benefiting our relocated species and neutral interactions with the microbes. And while this was a single plant, we definitely need more studies to generalize. But overall, this suggests that maybe we should consider these topographic relocations way before we consider these large latitudinal relocations. By moving a large distance of latitude, we're changing pollinators, we're changing herbivores, we're changing plants, we're changing microbes, we're changing a lot of potential species interactions. Through these smaller scale interactions, we're moving a shorter distance and changing fewer interactions. In our system, we probably have the same pollinators and same herbivores at our recipient site as we do as our home sites. Some of the same plants, but positive interactions and neutral interactions with the microbes. So this may really be a good way to start thinking about these managed relocations, is trying to do it in these shorter distances as possible. And with that, I'd like to thank the many people that helped with this project and our funding for the 2011 part of this project. And please feel free to leave any questions or email me or hit me up on Twitter. Thank you very much for listening.